Let's talk a little bit about protein substrate affinity, and that is the strength with which a protein binds to its particular substrate. Now that's important for a lot of proteins because they tend to bind to and modify their particular substrate, like an enzyme, or bind to and transport particular substrate, such as a membrane transport protein or the circulatory protein hemoglobin that binds and transports oxygen. Now, if we look at the graph in A here, we can see that as substrate concentration rises in the presence of our protein binding of that substrate should also rise. And we see that's true with our blue line rising as we go to the right here with increased substrate concentration. Now, how do we infer the protein affinity from this graph? Well, we can choose an arbitrary value, 50% saturation, meaning 50% of the binding sites of our particular protein in question are bound to the substrate, which means half of those sites are also unbound. What is the concentration of substrate required to do that? Well, if we read across from our 50% saturation and then down to our substrate concentration, we find that there's a particular substrate concentration that will provide that particular degree of binding. That is for this given protein under a certain set of circumstances. What we'll see in a moment is that that can actually change. Now, that value, that amount of substrate required to generate 50% saturation is called the dissociation constant. Right? And it is a measure of what is called affinity. And again, that's the strength with which a protein binds to its particular substrate. Now, if we go over here to B, we can see that in an unmodified protein, under, again, a given set of circumstances, particular environment, it demonstrates a certain affinity. Here it is. Right? If we go down from the blue line over from our 50%, right, we would get this particular KD value. Now, protein affinity can be modified in certain circumstances. In some cases, one mechanism is called allosteric modulation. So if we use hemoglobin as our example, hemoglobin binds oxygen at 4 separate locations shown here. Well, it also binds carbon dioxide at a couple other locations, well, 4 other locations. And the binding of carbon dioxide to those particular sites actually modifies the affinity of this protein for oxygen at these other locations. That's called allosteric modulation. Now, it can be either positive or negative. So if it's positive allosteric regulation or modulation, we end up with a left shifted curve and a KD value that is lower, right? Because it requires less substrate to generate the same degree of binding, this arbitrary 50%. Right? If you need less substrate to get 50% binding, it must have higher affinity. Oppositely, right, we could have negative allosteric modulation, in which case our protein will require more substrate to get that 50% binding, right? We go over here, come down to a higher KD. And what that tells us is that KD, our dissociation constant, is inversely proportional to affinity. In other words, as KD goes up, our affinity, what it indicates, is that our affinity is going down, the strength with which it's binding its particular substrate.